In this class, we will study the effects of water stress on crop production. This class is based on Chapter 14 of the book Principles of Agronomy for Sustainable Agriculture, published by Springer. The authors are Víctor Sadras, Francisco Villalobos, Francisco Orgaz, and Elias Ferreres. The title is Effects of Water Stress on Crop Production. First of all, what is the importance, the relevance of water stress in agriculture? Water stress is the main limiting factor for agricultural productivity in the world. So, the main aspects that we can improve for increasing agricultural productivity are related to water stress. Also, water stress is the main cause of tree mortality in many ecosystems. Apart from that, water stress has an effect and interacts with other environmental or plant factors. For instance, if water stress occurs, if water absorption is reduced, nutrient absorption is also reduced. Also, water stress, the lack of water for transpiration, affects the canopy microclimate, in special as it affects the canopy temperature. The definition of water deficit is the occurrence of a very low water potential in the plant. When we say low, we are referring that it is low enough for affecting physiological processes in the plant. Remember that the water potential refers to the energy per unit mass or volume water either in the plant or in the soil. In principle, we can write an equation to express the flow of water through the plant as proportional to the difference between the water potential in the soil and the water potential in the leaves. And this difference is divided by the resistance to water flow from the soil to the leaves. This equation of water flow through the plant uses an analog water flow with the flow of resistance in an electrical circuit. Using this equation, we may derive the value of the water potential in the leaf. Should be equal to the water potential in the soil minus the product of plant transpiration by the total resistance flow of water from the soil to the leaf. This is a very important equation because it tells us when we can have water deficits in the plant, when, under which conditions the water potential in the leaf is going to be low. Using this equation, it is clear that the potential in the leaf is going to be low, for instance, when we have a very low value of the water potential in the soil. And that occurs for when the soil is dry, and also when the soil has a high concentration of salts. Also, we will have a low leaf water potential when the plant transpiration is high. And this occurs when we have a high evaporative demand, in other words, high reference evapotranspiration, and also when we have a large leaf area. So, high evaporative demand, high leaf area lead to large transpiration, and therefore we will have a low leaf water potential. The third factor involved is the resistance to the flow of water from the soil to the leaf. And this resistance is going to be low when we have a low soil hydraulic conductivity that occurs in dry soils. Also, clay soils will have a lower water hydraulic conductivity than sandy soils. High resistance is also caused by a low root density. So if the root length density is low in the soil, we will have a high resistance to the flow. Apart from that, we can have a high resistance due to a high root radial resistance. This is the resistance to the flow from the soil to the root. And this occurs typically, for instance, when the soil temperature is very low. Apart from that, any factor inducing a high resistance in the xylem of the plant, for instance, due to a disease, will increase the resistance to the flow from the soil to the leaves. Let's remember that we can always decompose the water potential in any part of the plant among different components. The first is the matrix potential, which is associated to the absorption of water to solids either in the plant or the soil. The second component is the pressure potential or turgor potential. This is a positive potential that enables, for instance, plant growth. The gravitational potential is usually negligible in the case of agricultural plants. It's only important when we have very, very tall trees. And in the fourth place, we have the osmotic potential that is associated with the accumulation of solutes in the cells of the plants or in the soil solution. This is proportional to the amount of solutes that we have in the solution. For instance, inside the vacuole, a total water potential of minus 0.5 megapascal might be associated with 0 matrix potential, 0.2 turgor potential, which means that the vacuole is going to be exerting pressure, positive pressure, 0 if we put in this place the reference surface, so we will have 0 gravitational potential, and finally minus 0.7 osmotic potential. So we see that the osmotic potential within vacuoles can be important. In the case of the cell wall, the osmotic potential is very low. We have not much solids at the cell wall. We have 0 for gravitational, 0 for pressure potential, and we have now an important matrix potential. And this would mean that we have an equilibrium, so water would not be moving from the vacuole to the cell wall or vice versa. It is interesting to point out that during the day we have usually a dehydration of the plant. That occurs because the curve of transpiration and the curve of water uptake are delayed in time. Transpiration starts sooner than water uptake, and in fact transpiration causes the gradient of potentials that is later responsible for water uptake. By looking at these curves, we see that during part of the morning the plant is losing water, and during part of the afternoon the plant is regaining water. Usually during the night the water uptake, we will have 0 transpiration or almost 0 transpiration, and water uptake during the night will refill the water capacitance of the plant. in this figure we see a simple diagram for the soil plant atmosphere continuum. Usually we talk about the SPAC, soil plant atmosphere continuum, that is related to the flux of water from the soil to the atmosphere. The plants act simply as a channel of water from the soil to the atmosphere, and the amounts of water that flows through the plants is much more important than the total water that is contained in the plant at any time. You see here different resistances, starting with a soil resistance. This is the resistance for flow from the matrix of the soil to the surface of the root. Then, when we arrive at the root surface, we will encounter an important radial root resistance. We put here an arrow indicating that this resistance is variable. We will talk about this later. Once the water has been gone into the root, we will find an axial resistance in the xylem of the roots, then in the xylem of the stem, and then it will arrive to the leaf, where it will have two resistances in parallel. One, that of the cuticle, and one due to the stomata. Look that once again the stomata present a resistance which is variable. It will be low when the stomata are open, but it will be very high if the stomata close. In general, the resistance due to the cuticle will be very high, so water will flow usually through the stomata from the plant to the atmosphere. We said before that the resistance of flow of water from the soil to the atmosphere depends very much on the root length. So if the root length is very high, then the resistance to the flow will be low. Here we see a comparison of leaf area index, LAI, and total root length for cereals and dicots, in this case legumes. We see that the total root length of cereals is much higher, even for lower lies, than that for dicots. In other words, the amount of roots, mostly in the upper layer of cereals, is very high as compared to that of dicots. This is probably not required for water absorption, but for the absorption of other nutrients like phosphorus. We said also, by looking at the diagram, that we have a variable radial root resistance. This is due first to the fact that the water flow has to go through membranes. But these membranes have a special type of proteins called aquaporins that may enhance greatly the flow of water through the root surface. Aquaporins can control this water flow across the membranes by changes in the abundance of aquaporins, so we will have more doors for water entering the roots, or by opening and closing the channel. It has been observed that root resistance may change during the day in association with changes in the abundance of aquaporins. The main effects of water deficit on crop productivity may be explained using the general equation of crop yields that we have already seen in previous lectures. This equation tells us that yield is the product of three factors, namely radiation intercepted, radiation use efficiency, and the harvest index. Remember that the harvest index is the fraction of total biomass used for the harvestable yield. Well, the limitations to yield due to water stress are due to, first, reduced expansion growth. The crops will have a lower leaf area index, so they will intercept smaller radiation amount. The second factor due to water stress is stomatal closure. That causes reduced photosynthesis, reduced plant transpiration. In other words, the radiation use efficiency will be reduced due to stomatal closure. When the water stress is severe enough, other factors may be affected too, for instance, pollination or seed set, and that has an effect on the reduction in the harvest index. In summary, the yield may be reduced by water stress due to reductions in the radiation interception because of lower growth, reductions in the radiation use efficiency because of the stomatal closure, or reductions in the harvest index due to poorer seed set. Usually, the reductions in harvest index require a very severe water stress. Let's review these different factors that affect productivity. In the first is growth. The relative expansion rate of the leaves of the crop might be written as this: the increment by unit time divided by the volume of an organ is proportional to a constant, which is the cell wall extensibility, multiplied by the difference between turgor pressure, or pressure potential, minus a threshold value of turgor pressure. The value, the threshold value for increases for expansive growth is rather high, which means that if pressure potential is reduced, we will readily achieve zero relative expansion growth rate. So remember, the values of threshold pressure for growth are rather high, so growth is very sensitive to water stress. In the case of stomatal functioning, that we said that was the second factor affecting productivity, first of all we have to agree that it is a very complex process and is highly dynamic. The stomata might be opening or closing in terms of seconds or minutes, and the stomatal opening is affected both by environmental and plant factors. Environmental factors important for stomatal closure or opening are the concentration of CO2, the leaf temperature, the vapor pressure deficit around the leaf, the radiation amounts, and the light quality. But we have also plant factors affecting stomatal opening, like leaf age or the source-sink ratio. In other words, the concentration of sugars in the leaf may reduce photosynthesis through the stomatal closure. The first, more important factor affecting stomatal closure is the turgor pressure potential of the guard cells of the stomata. If this turgor pressure is high, we tend to have open the stomata. If the turgor pressure is low, we tend to have closed stomata. In any case, the main factors that we can find in most agricultural species are high BPD tends to close the stomata. Root signals associated to dry soils or compacted soils tend to promote stomatal closure. Low radiation tends to promote also stomatal closure. And also high CO2 concentrations promote stomatal closure. These mechanisms are complex among other causes because we have an important feedback loop here. If we have a low leaf water potential, if we have water stress, that promotes stomatal closure because the turgor pressure in the guard cells is going to be low. But if the stomata close, then we will reduce transpiration. Therefore, if we reduce transpiration, then the potential will tend to increase, which means that the stomata will tend to open a little bit. So we have a feedback loop between water potential and stomatal conductance. But the stomata are not only responsible for regulating the water use efficiency, which means that by closing the stomata, we avoid losing water, but we also stop CO2 from arriving for photosynthesis to occur. There are other roles of the stomata like avoiding silent cavitation and dehydration of the plants. This is very important for survival of the plants in many situations. But it is also important to remember that transpiration regulates canopy temperature. And by transpiring and opening the stomata for transpiration, the plant cools in comparison to the outside temperature. The relationship between the stomatal conductance and water potential in the plant depends on the species, but also we can see differences between cultivars of the same species. In general, we have said that the stomatal conductance declines as water deficit develops, for instance, if the soil dries. But see, in this case, for vines, we have two different cultivars, Grenache and Syrah, and we see that Grenache controls more strongly stomatal conductance as the soil dries than does Syrah. The case of Grenache is closer to what we call isohydric behavior. In the case of Syrah, we are closer to the so-called anisohydric behavior. Isohydric means that the plant tends to keep or avoids reducing its water potential by closing stomata sooner. In the case of anisohydric plants, the plant has a less strong control of conductance due to water potential. In other words, anisohydric plants will allow much lower water potentials in the plant. We said before that the stomata do not only regulate the entry of CO2 or the exit of water, it also controls the temperature of the crop. Here we see the results of one experiment where we had irrigated Shiraz vines that were grown under control, the dark symbols, or heated plants. These are the heated plants. And we have a comparison during the morning or the afternoon of stomatal conductance, transpiration, or photosynthesis. In general, all variables increase when the plants were heated. The conductance was increased in the morning and in the afternoon, and that allowed higher transpiration in the morning and the afternoon. Photosynthesis increased just a little bit, both in morning and the afternoon, due to heating of the canopy. The point here is that in all the cases when the plants were heated, the evaporative cooling due to transpiration allowed that not more than 1 degree Celsius temperature was observed between the heated plants and the control plants. In other words, the control of the temperature by increasing transpiration was more important than maintaining the water use efficiency of the plant. So, by opening the stomata, the transpiration was higher, which cooled the plants, avoided high temperatures, but reduced the water use efficiency. So, the idea here is that keeping the canopy cool is, under some conditions, more important than saving water for the crop. We will see here an example of observations of canopy temperature. We have here an experiment where we have a control well-irrigated tree in the center. To the right, a deficit irrigated tree which had around 40% of the water as the control, and to the left we have a tree that was treated using kaolinite, which is a white powder that increases the albedo of the plant. In the back we have a wet cloth to increase the thermal contrast between the plants and the surroundings. Here we see Dr. Álvaro López Bernal controlling the infrared camera using a laptop, and here we have the trees, and this is an afternoon during September 2014. Here we see a thermal image. We see in the center, remember that we have the control tree, which is colder than the water deficit tree. To the right, the yellow or red indicates a higher temperature, while the blue indicates cooler temperature. We see here that the center of the tree is hotter than the tips of the stems of these olive trees. The behavior of stomata is further complicated by the existence of stomatal oscillations. These oscillations have been observed for many species, including olive trees, under clear sky conditions with no apparent reason. They are usually more important in small trees during the summer, and they are quite frequent in citrus species like orange, also in large trees. Here in this figure we see an example of the occurrence of stomatal oscillations in the case of olive trees in summer 2014, and we are here showing the transpiration subflow measured using the subflow sensors of 3 different trees. The black line shows a control well-irrigated tree, while the red and the blue show trees under deficit irrigation. We see that during most of the day, the control tree doesn't show oscillations, but oscillations start soon after noon. And the amplitude of these oscillations is increasing. But the oscillations are stronger in the case of tree D1, which is a deficit irrigated tree, where oscillations start soon after 10 o'clock in the morning. And the amplitude of oscillations is very, very important. Look at this. The transpiration goes from 0.1 up to 0.65. And these periods are around 50 minutes, which means that during around 1 hour, the stomata go from practically closed stomata to fully open stomata during most of the day. The other deficit tree shows also strong oscillations, not as important as the red one. Note that this was a typical summer day with clear skies, so there is no apparent reason for these very strong oscillations. Okay, now we will see how the crop regulates transpiration. To analyze that, we will decompose transpiration among three different factors, which are leaf area index. The second factor would be intercepted radiation per unit leaf area index, and the third would be transpiration per unit intercepted radiation. The first factor, leaf area index, is modulated by the tillering rate, branching rate, the leaf growth rate, or the leaf senescence rate. So, by producing more tillers or branches, we can reach a higher leaf area index. By increasing the leaf growth rate, we have a higher leaf area index. By increasing leaf senescence, we reduce the leaf area index. This is the main regulator of transpiration of crops, changes in leaf area index. The second is the amount of intercepted radiation per unit line. That depends on the canopy architecture, namely the leaf angles, which can also be affected by water stress, as we will see later. Under conditions of water stress, we will typically have less tillering, less branching, lower leaf growth rates, and higher leaf senescence rates. Also, under conditions of water stress, in many situations we have less intercepted radiation per unit line. The third factor, the amount of transpiration per unit intercepted radiation, is associated with stomatal closure. So, as we said before, plant growth is very sensitive to water deficits. So, the main regulator of transpiration due to water stress will be reduced leaf area index. In the second case, we will have effects through canopy architecture and stomatal closure. Here we see the results of experiments with sunflower performed in Cordoba in spring or summer, where we measure leaf expansion and transpiration of the plants for different values of soil water content. Here 1 means field capacity, while 0 means permanent wilting point. We see that the leaf expansion during summer will start to decrease very soon after we depart from field capacity. If we are in spring, the expansion will not be affected unless a much higher amount of water has been taken from the soil. Transpiration, on the other hand, is less sensitive to water stress, so it will not be affected until around 70–80% of the soil water has been used by the plant. Therefore, for the same water content, we have more sensitive expansion during summer than during spring. And this is explained by the effects of leaf water potential on the on expansion. Here, in this table, we summarize the relative importance going from greatest, 3 crosses, to negligible, minus sign, of different mechanisms that regulate transpiration of sunflower in pre- and post-anthesis. The process that we consider are first, intercepted radiation, second, amount of transpiration per unit intercepted radiation. If we reduce intercepted radiation, this may be due to reduce leaf growth rate or to increase leaf senescence. But we can also reduce intercepted radiation, reducing the extinction coefficient, which means wilting of the plant. Also, we can reduce the transpiration per unit intercepted radiation by closing the stomata, by reducing stomatal conductance. The results of these mechanisms are reduced leaf area index, either by decreasing leaf growth rate or increasing the leaf senescence rate. By reducing the extinction coefficient, we will reduce the amount of intercepted radiation per unit leaf area, and by reducing stomatal conductance, we will reduce the amount of transpiration per unit intercepted radiation. We see that reductions in the leaf area index are very important in pre-anthesis and not much important in post-anthesis. Why? Because most leaf growth occurs pre-anthesis. Leaf senescence has a stronger effect in post-anthesis than in pre-anthesis. So, in pre-anthesis, we change leaf area index by changing growth rates, and in post-anthesis, we change the leaf area index by accelerating leaf senescence. On the other hand, the effects on the extinction coefficient are more important in post-anthesis than in pre-anthesis. And finally, stomatal closure has a stronger effect in post-anthesis than in pre-anthesis. So, in summary, the regulation of transpiration in the case of sunflower, in the case of during pre-anthesis, is associated with leaf growth rate, and in post-anthesis is regulated by leaf senescence, stomatal closure, and reducing the extinction coefficient. Water stress has other effects on the plant associated with other factors that limit productivity. We talk about a water nutrient co-limitation because when we don't have uptake of water, we won't have uptake of nitrogen. That occurs in special for cereals in Mediterranean environments for the case of nitrogen. We have also the amplifying effect of water stress on heat stress. If the plant does not transpire, it will heat more. So, under conditions of heat stress, the effects of heat stress will be stronger on the plant. We have to remember here that the interactions between water stress and other factors may be complex. So, for instance, nitrogen fertilizer may be positive or negative when water stress occurs. For instance, in this table we see results of different experiments performed in Australia with different nitrogen rates from non-nitrogen applied low fertilizer applied, high fertilization rate, using wheat with half density or normal density. These are results of rain-fed crops. In this case, we are here the table reports the percent damage of the ears of the crop due to heat stress, and we see that the heat stress was more harmful when we had normal density with high nitrogen applied. When we applied no nitrogen and used only half density, we had the less damage to the crop. What is the explanation for this? If we increase density and we improve the nutrient status of the crop will use faster the water, so less water will be available during grain filling. If we have not much water during grain filling, water stress is going to be severe, and the plants under heat stress will be hotter, so the effect of high temperatures will be more severe under these conditions. If we have less plants in the field and we don't apply nitrogen, the plants will use water more slowly, so more water will be available during grain filling, and the crop will transpire more and will cool more under these conditions than under these conditions. In this case, the same experiment was performed under irrigation, and in that case the heat stress had no effect on the ear. There was no heat damage to the ears of irrigated crops. One important question is if susceptibility to pests and diseases is affected by water deficit. And in general, positive and negative effects have been observed. For instance, mites in cotton prefer irrigated plants because water-stressed plants show thicker leaf cuticles. Irrigated plants have thinner leaf cuticles and are preferred by the mites. In the case of root diseases, the water stress will have a stronger effect, will have a more important effect, because the root resistance to water flow will be affected. The main effects of water stress when stomata close are related to the stomata being the door for CO2 entering the plant and water leaving the plant in transpiration. We can put together the equations of flow for transpiration and net photosynthesis, which are the two a function of the stomatal conductance. Transpiration is equal to 1.6 stomatal conductance by the difference in vapor pressure between the substomatal cavity and the atmosphere divided by atmospheric pressure. Photosynthesis is equal, or may be written as, the product of stomatal conductance multiplied by the difference in CO2 concentration between the atmosphere and the substomatal cavity. Now we do the small trick of writing the difference in vapor pressure by adding and subtracting the vapor pressure at saturation at air temperature. We see that this difference in vapor pressure between the substomatal cavity and the atmosphere is equal to vapor pressure deficit plus delta multiplied by the difference in temperature between the crop and the atmosphere. Delta is the slope of the saturation vapor pressure curve versus temperature. Now we can write the ratio between CO2 flux entering the leaf, and transpiration, water vapor flux leaving the leaf. As this, we see that we eliminate the stomatal conductance, and the ratio between photosynthesis and transpiration, also called water use efficiency, is equal to atmospheric pressure P multiplied the difference in concentration, CO2 concentration between the atmosphere and the substomatal cavity divided by 1.6 BPD plus delta difference in temperature between the crop and the atmosphere. If we assume that the difference in temperature between the crop and the atmosphere is negligible, just for simplifying this analysis, we can write that Pn over transpiration, photosynthesis over transpiration, and water use efficiency is equal to Ca 1 minus Ci over Ca. This is the ratio of CO2 concentration in the substomatal cavity and the atmosphere, multiplied by atmospheric pressure and divided by 1.6 BPD. If this ratio of CO2 concentrations is constant, and many researchers have observed that this is between 0.7 and 0.8 for C3 plants and between 0.3 and 0.4 for C4 plants, and is rather constant in most situations, we can write that the water use efficiency is equal to a constant divided by the vapor pressure deficit. This is a very important relationship that tells us that the water use efficiency decreases as the environment gets drier or hotter. Water use efficiency decreases as BPD increases. Also, in this equation, we see that the water use efficiency increases if the atmospheric CO2 concentration increases. So, we could wonder, with global change, we are increasing the CO2 concentration of the atmosphere and the climate is getting warmer. What will be the effect on the water use efficiency? Because increased CO2 tends to increase water use efficiency, but increased temperature tends to decrease water use efficiency. Another question that we may ask ourselves is that if we can increase the water use efficiency simply by plant breeding, is it possible to have plants that can get CO2 without losing water vapor? To show that this is not just a theoretical consideration, we see here a Univ covariance system on cotton crop in summer in Cordoba 2003, and this system allows measuring the fluxes of CO2 and water vapor over this plane. Using this data, we can calculate the ratio of CO2 arriving to the crop over the amount of vapor pressure that is leaving the crop. This is the ratio of canopy photosynthesis and evapotranspiration. We can also call that water use efficiency. And see what happens. These are data during the daytime, during the summer 2003, and we see that the water use efficiency, the amount of CO2 that we get per unit of water lost, decreases as the theory predicts as we increase the vapor pressure deficit. The equations that we have seen may allow calculating biomass as a function of transpiration. So we write that the increase of biomass of a crop or a plant is the product of the transpiration by the water use efficiency. And we see that the water use efficiency is a function of a constant that depends on being C3 or C4 and the vapor pressure deficit. This constant is between 3.1 and 4.6 for C3 plants using a current concentration of CO2 of 397 parts per million. For C4 plants, we have a constant between 9.3 and 10.9 grams of dry matter per liter multiplied by kilopascal for the current concentration CO2 of 397 ppm. We can apply those equations to calculate water use efficiency of crops in Cordoba. For instance, for wheat planted in December, harvested at the end of May, we apply the equations that were previously presented with a seasonal BPD of 0.7 kilopascal. We come up that we can expect 5.6 grams of dry matter water use efficiency per liter of water transpired. For olive trees, with a seasonal BPD of 1.2, we come up with a water use efficiency of 3.2 grams of dry matter per liter of water transpired. For sunflower, going from February to July, mean vapor pressure deficit of 1.3 kilopascal, we come up with 3 grams of dry matter can be accumulated per liter of water transpired. Water stress has other effects on the plant. In general, in agricultural plants, mortality is not much affected by water stress. But the reduced under severe water stress, the reduced rates of cell division and expansion translate into abnormal cell differentiation, and abortion of flowers, and or reduction in flower primordia or fruit set. That means that, in general, the severe water stress affects harvest index. The hydrolysis of proteins due to water stress and the accumulation of amino acids might reduce directly CO2 fixation. Also, we have changes in some hormones, for instance, increases in abscisic acid and ethylene that accelerate leaf senescence. This is also associated to reductions in indolacetic acid, cytokinins, and gibberellins. Water stress also reduces nutrient uptake, but has little effects on respiration, on the transport of assimilates, and on the rate of development. As we said before, it might affect the susceptibility to pests or diseases. And how plants cope with water stress? Well, one interesting possibility is to avoid completely the water stress if the plant has a growing cycle that is tuned to the water availability. But in some cases, the crop grows during periods where water stress cannot be avoided. So, in some cases, we have a higher tolerance to water stress by different mechanisms, usually related to reducing the demand for water or increasing the water supply to the plant. And that may be achieved by reducing the leaf area index or increase the root length, by reducing the radiation interception, changes in leaf angle, rolling of the leaves, temporal wilting, or leaf losses by senescence. We can also reduce the stomatal conductance by stomatal closure or increasing the thickness of the cuticle of the leaf. Another way of promoting plant tolerance to water stress is to reduce the impact of stress on the plant mechanisms. And one important way is the osmotic adjustment, which means accumulation of sugars, amino acids, potassium, that keep pressure potential for growth of the plant and to keep the stomata open. We see here an example of changes in the architecture of the plant due to water stress. These are maize plants that show leaf rolling and more vertical leaves, which reduces radiation interception. A similar response is observed in a water-stressed wheat crop that shows also leaf rolling and more vertical leaves. In some fruit trees, what will occur under water stress is fruit dehydration. This is the case of olive trees. Under severe water stress in olive trees, we will observe turgor loss. Look at the tips of the stems are curled, have lost turgor. We observe also leaf curling. The leaf tends to roll, and changes in leaf angle. The leaves tend to be more vertical. If the water stress is more severe, we will observe the death of leaves or even the death of full stem sections. To quantify the impact of water deficit on crop production, we might use the simple equation of Stewart or more complex crop models. The equation of Stewart is basically based on the relationship between yield and transpiration. And manipulating these equations, we come up with this, which is the so-called Stewart equation that states that the yield loss due to water deficit, which is this relative loss, is proportional to the relative reduction in evapotranspiration. Here yield, Y means yield, and ET means seasonal evapotranspiration. The proportionality coefficient lies between 0.6 and 1.5. Depends on the crop species and also depends on the partitioning between transpiration and soil evaporation. For instance, for a maize crop with a potential yield of 12 tons per hectare and maximum evapotranspiration of 700 millimeters, if the proportionality coefficient is 1.4, with a seasonal evapotranspiration of 400, we will produce only 4.8 tons per hectare. However, the equation of Stewart is quite limited. It will not be valid if we have a lot of variations in water use efficiency, if we don't know a priori the maximum yield, or if we change the irrigation method, and it's not valid for conditions of severe water stress when we have acclimation to water stress. Finally, we could use alternatively for calculating productivity under water stress conditions the model aquacrop, which was developed by the Land and Water Division of the FAO and includes different utilities for managing climate data, crop data, different types of irrigation, allows calculating crop water requirements or irrigation dates, but also allows the user to calculate the impact of water deficit on crop yields.